everybody, according to my glycolysis, I will use this book, Biology Today. So, but I will simplify the information inside. As I will use during this presentation. So this is ATP, which contains adenosine, triphosphate. Adenosine is the yellow ball, and the triphosphate are the pink balls. And when it is dephosphorylated, so or hydrolyzed, it will give ADP and phosphorus. So then we'll obtain the AGP. And here NAD is the orange ball, and NADH is the orange and blue ball. So we suppose that hydrogen would be the blue ball. Here we recognize that we have glucose, which is 6 carbon molecule, is converted to 3 carbon molecule, which is called pyruvate. But here the 6 carbon molecule will give 2 pyruvates. Okay, so here we have glucose plus 2 ADP plus 2 phosphorus plus 2 NAD will give 2 pyruvate, 2 ATP, 2 NADH. Okay, why we multiplied by 2? Because the glucose molecule is 6 carbon atom, and we will see during the process it will be split into 2, and we will continue with only 1 3 carbon atom till the end. So we will multiply the result by 2. Okay, this is part 1. Okay, so this is in brief. Okay, I will talk about it here later. This is part 3, part 2, part 3, until we reach the pyruvate. Now I will start with part 1, okay, because we have part 2 and part 3 as well. Part 1 here, I will start with glucose molecule, which is consists of 6 carbon atoms, and here we will use magnesium and hexokinase, which is an enzyme that is responsible to release phosphorus or add phosphorus. So here this is the ATP, okay, it is entered as ATP. So here in this process, part 1, we consume ATP, so we will consume 2 ATP through this process, okay, through part 1. And then in part 3 we will get 4 ATP. Okay, so here we will remove this phosphorus and add it to phosphorylate the glucose, so we will obtain glucose 6-phosphate. After that we will have an enzyme here, is called isomerase, which is phosphofructo or phosphogluco isomerase. So it's okay, they are the same. Here we will obtain that it is phosphorylated, as you see here, and the name become fructose 6-phosphate. Why we convert it to fructose 6-phosphate to its isomer? Because here fructose 6-phosphate is it is more symmetric, so it prepares the glucose to be cleaved later into 2 identical halves. And here we will observe that by using magnesium and using phosphofructokinase, here again we will remove this phosphorus, okay, and add it to this molecule to obtain fructose 1,6-biphosphate. Okay, this is the end of part 1. So here after that we will start by adding magnesium and fructose biphosphate aldolase to cleave this compound into 2. Okay, this will be in part 2. I will start part 2, where we cleave the 6 carbon atoms to give 3 carbon atoms. Here 2 carbon atoms, 3 and 3, 2 molecules, each one has 3 carbon atoms. So here in this process we will start by, in part 2 we will start by fructose 1,6-biphosphate here, by using fructose biphosphate aldolase and in the presence of magnesium as a catalyst. Then we will obtain that it will cleave these 6 carbon to give dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which is the GAP, okay? And here by the isomerase enzyme they are converted to each others, okay? This is happening in part 2. And also in part 2 we will obtain that we have here glyceraldehyde 3-phosphate dehydrogenase, okay? Here it has in the absence of the NAD, here we will need a NAD molecule, okay? And here this phosphorus
would be added later, okay? And here we will use this NAD molecule, which will then gain by the dehydrogenase. It will gain a hydrogen and will give this molecule. And here the phosphorus will be added here, okay? So we'll obtain 1,3-biphosphoglycerate. Okay, so here we will start part 3. Part 3 will start from here. 1,3-biphosphoglycerate. In this process, part 3, we will get the goal of the glycolysis, which is the release of ATP. So we'll obtain 2 ATP through this process, but at the end we will multiply it by 2, so we will obtain 4 ATP. But in the beginning here we already consumed 2 ATP, okay? So we will subtract at the end 4 ATP from the 2 ATP to obtain 2 ATP as a final result. So here you will find that by using phosphoglycerate kinase, okay, this will get 1 phosphorus from this compound, dephosphorylation, okay? So after that we will obtain this ATP through this step, part 3. This is our first ATP which we will obtain. Okay, so now we got 3-phosphoglycerate. By using the enzyme phosphoglycerate mutase, it will convert this phosphorus, it will move it into carbon 2. So instead of being in carbon 3, it will be in carbon 2. Okay, and here we will get, by using the magnesium and enolase, we will get the phosphoenol pyruvate. And by using the pyruvate kinase, okay, so that will remove the phosphorus here. So we'll get the second ATP through this glycolysis, and then we obtained the pyruvate, okay? But we will obtain only 1 pyruvate through this process, okay? But we'll multiply it by 2. Why? Because the origin of this process was a 6-carbon molecule of the glucose, which will then was cleaved into 2 carbons, and the majority of the 3-carbon molecule here is converted to glyceraldehyde 3-phosphate. So this one will continue the process, okay? And here we will get the 2 pyruvate at the end, okay? So we will multiply the NAD here by 2, and the ATP here we will multiply it as well by 2, and here the other ATP will multiply it by 2, okay? So now we have 4 ATP from here, 2 from here, and 2 from here, okay? And we will subtract those 2 ATP which we started with here in this part 1 process. But here we will not multiply it by 2 because the 6th molecule as a bulky whole. So it's okay, we will subtract the 4 ATP molecule from these 2 ATP molecule to obtain the final result, which is the 2 ATP as you see here. So thank you very much for watching, and I hope that I simplified the glycolysis process. Thank you.